It's really great to be able to talk to you a little bit about and this opportunity to tell everybody here what we've been up to and why this may be important to those studying diabetes and obesity. Our lab's kind of been spending a lot of effort over the years in trying to make it so that we can get more robust quantitative measurements of proteins by mass spectrometry. And we've worked with a number of different collaborators. And one of the things that I think has been pretty exciting is that in the last couple of years, we've worked specifically with the Hoofnagel lab to apply some of these tools and methodology towards developing quantitative methods for diabetes and obesity. So one of the things that obviously becomes to be a challenge is you can have a lab that's extremely specialized and has a lot of expertise in these very complicated and expensive instrumentation. But unless you can actually share those methods and other people can disseminate it and get them up and going again, it's less powerful. So a major effort, what I'm going to talk about is the tools that our lab has kind of developed to try to take these extremely complicated pieces of instrument hardware and make it so that we can transfer methods between labs, et cetera, and get reproducible results. So I titled this talk as PanoramaWeb. A lot of this will be around this web application we call PanoramaWeb. And we think of it as kind of like a knowledge base or a resource to manage, share, and collaborate with these mass spectrometry experiments. The website is this panoramaweb.org. Our lab website is MacCosslab.org if people have any interest to follow up on. So a little bit to kind of get a little bit more details here is when this got started was around about 2006 or so. We started to think and realize that everybody had different instrument platforms, and what we were trying to do was all the same, and that is we need to be able to take a protein that we wanted to make an assay for, and we need to be able to generate candidate peptides that we're going to be able to quantify. And we need to be able to build that into the instrument. And we need to be able to work across all major instrument vendors. That was actually not that simple of a problem. And so we started building this client tool, which we call Skyline. And this is kind of where PanoramaWeb all got started. It was a client tool. It's a Windows app. And it's basically a document editor, so we think about this as being a tool in which somebody could actually build methods. They could apply those methods on the instrument and analyze the data and visualize it and adjust the results from it. But just like all document editors, just like Word or Excel, you spend a lot of time optimizing these methods. So you need to have things like multi-level undo, redo, and that's something that works really well in a client tool as opposed to a web application. It allows you to adjust the method, the end results, and these methods and these documents get very complicated. And we also wanted to, all the other existing tools use kind of these vendor neutral file formats. We wanted the ability to read the vendor formats natively without having to deal with file conversion. This was a fairly complicated process to deal with very large companies that have these proprietary formats. And we wanted the ability to make use of prior data. So there were a lot of resources within the mass spectrometry and proteomics fields. We want to be able to build on what we call spectrum libraries if we could. We wanted the software to be very transparent, so free and open source. There were vendors that were selling very expensive pieces of software, and what would happen is that there would be a piece of software on one instrument computer or one lab computer that then everybody would take their turns to use, and we wanted something where everyone can install it on every computer in the lab and make use of it. And we wanted it to use professional development practices, and this is something we wanted it to be, extensively tested. We wanted it to be developed and shared in multiple languages. I won't talk too much about that, but we wanted to have things like true audit tracking and audit logs, et cetera. And so I'll talk a little bit more details in this, but this is basically what the application looks like. It's one of the simpler type views. And we support now these 6 major instrument vendors. And one of the things I like to say is that not only do we support their platforms, these actually instrument vendors have now come to be quite a close working relationship where they actually support our project as well. So after we were using Skyline for a while, and we started to see Skyline being used in thousands of laboratories. I would go into someone's laboratory, and I would see that somebody would have like this document structure, these series of like nested folders on their file system, and they would store all their documents and their raw files in kind of some their own general way. And people were constantly trying to find the right document that they could use to show me something within their software and their results. We felt like this would be much more useful if it could be stored within a relational database. We thought that people could do queries with it. And we wanted to build the software tool we call Panorama. We did this with a company in the Seattle area called LabKey Software. It's kind of a spin out of initially of the Fred Hutchinson Cancer Research Center. It was initially designed for sharing these Skyline documents and the associated data. We actually host a free version of the software, which is we call PanoramaWeb. There are now 574 independent labs that have accounts on it. There's been over 24,000 Skyline documents uploaded, and I'll talk more about this in a second. Basically, each lab is given an account. There's an administrator for that lab account, and they control the security of their documents and all the subsequent folders that they have up there. We found it to be an excellent mechanism for sharing data between labs and also the public. We've started to notice that several of the large NIH-funded programs have started to make use of this. And this has been fantastic to see. We've had a number of different papers on this. In fact, it's been kind of highlighted in a lot of the journal recommendations for sharing mass spectrometry data, that they actually place their data for transparency here within PanoramaWeb. We also, about 3 years ago, started what we call Panorama Public. And Panorama Public is basically part of the Proteome Exchange. And the Proteome Exchange, shown here on the right-hand side, is kind of this consortium of 7 different servers where we make the data findable, right? So upon publication, the journals require that you get a Proteome Exchange ID. The data is put up there, it's stored, it becomes immutable to the user, and you get reviewer accessibility to analyze and visualize the data, and then that data is then made public upon publication and is controlled. And so if you have data that's got quantitative results, most often people put that in a server like Panorama Public, and we control and handle that security. There's now about 330 public data sets. There are a lot more in review. So this is obviously kind of that we're at the kind of the hockey stick of this takeoff of that portion of it, and we're finding more and more people who are using Skyline documents, finding that Panorama is an easy way to share this. I will point out that it's not just our own server. We actually make the software fully available. People can install it locally. We've had people install it on Amazon Web Services and within their own intranets. We're not expecting everybody to use the server that we main and host, but we do host a version because we do know there is kind of a barrier to adoption, but we also want to make sure that people are aware that they actually can install it locally themselves. One of the key things we wanted early on was it to be very easy to interact with this from the Skyline client. So if you have a document and if you're about to go home in the evening, it's very easy for someone to post it to their folder in PanoramaWeb or any Panorama server. You can browse it just like you would your local file system and post these files. We completely version control them. We change so someone can be making changes to the document, and every new version and the changes made to it are stored and can be reverted after the fact. So this tool Skyline is this way in which people can build methods. It interfaces with the instrument computer. You can collect and visualize the results from those data. It basically becomes the processed quantitative results. These data then gets published to Panorama, which is a relational database There is a fully detailed web application that people can use to browse that data. There is extensive APIs that people can use Python or R or even just Excel in order to be able to analyze their data at any sort of level in which people would like to, or JavaScript. They can interface with their data. And then you can download those documents and interact with them again later in the Skyline document. You can share them with other people, and it can be, we use this a lot for kind of consortia where people are developing and sharing instrument methods. So we've had a lot of growth in this. As I said, we basically had almost this linear growth since we started this project in 2012 with the number of labs that are using it. We've also had I would say an increased adoption in people using it to store different Skyline documents. Labs that are using it now more frequently. I don't have a figure to show that for you, but we've definitely done those sorts of analytics. We have lots of different types of documents we can store, those that are kind of quality control type data, system suitability metrics, we call libraries, and also just experimental data sets. But the total number of documents has definitely been increasing quite dramatically. So just to kind of give you a sense, I showed you that rich client tool that we have. You can reproduce some of those within the web app, but not to its full extent. It's not as interactive, but it still has a lot of the results that are there. You can visualize a specific protein there, and you can look at the different peptides you want to measure. You can have different annotations you store with it. And all of these tables are completely configurable. You can adjust them. You can change what's displayed or not. You can look at a specific protein where the peptides are being measured under that protein. You can rank all these peptides for that protein by different criteria, like the retention time or the mass. And you can look at them based on their abundance. So a lot of the visualization capabilities we've normally put into our client tools are also available in this web app. And you can drill down deeper and look at some of the quantitative data. You can actually see the underlying data for a specific peptide and how that quantity is reflective back of a specific protein. We show it in kind of this chromatogram view, but also these bar plots, and we can kind of, aggregate these things based on the metadata that's there, like disease or healthy, et cetera. One of the things that's very important for those that are working in a regulated environment, our lab is not working in a regulated environment, is that everything that happens to that document has to be tracked. We track this in an audit log. So anything that gets done and happens to that document, whether or not it's changing an integration boundary, there's a full record of what was done. You can revert anything because we have multi-level undo. You can also see who did it. So often documents get passed between multiple different computers, and you can see on what computer and who the user is that actually performed that audit, that modification to the document. I'll also point out, because our documents used internationally in China and Japan and many different countries, we actually need to be able to display this in whatever the native language is. So depending on the language of the native computer, we actually have this fully internationalized. So if someone is using their computer that's using Chinese language, it will be displayed in Chinese. If it's actually if that document is sent to us, and we open it on a computer that's using the English language, we will then display this full document in the appropriate language. And this is pretty important, and allows us to be able. Same thing with all of our error handling. We display that in a fully internationalized way. We also display this audit log all on the server side. So when you upload a document, that's all tracked as well, and it becomes basically a permanent record of that results. And we're getting very close to what we call this what was known to be this FDA 21 CFR Part 11 compliance. Again, the full details of everything is stored in tables within the Panorama server. One of the key things in sharing an assay is part of the validation that goes into it. So some of that validation of a quantitative measurement is the linearity of the measurement. So we have, for instance, the known analyte or expected analyte concentration with the measured signal in the mass spectrometer. You can see the underlying peak areas in a bar plot, but you can see the standards, and there are unknown values that are then being extrapolated to a concentration, and there are QC metrics that are being reported as well. And this has been very powerful for people to then share the validation at the time of publication. And part of the more recent project we have actually with the NIDDK, Andy Hoofnagel had mentioned to me he really wanted the ability to share some extra validation statistics like within and between day precision. So each of these individual sets of box and whisker plots are shown here. The different colors of the dots are different days that the measurements are being made. We show then the within, the total variance, the coefficient of variance, the within day and between day variance. And we also then show the individual results. So on a given day, an individual row has all the individual measurements, and then each of the columns are the results from that measurement on a given day. So this is an example of an assay that was being developed by the Hoofnagel lab here at the University of Washington. We also then track a fair amount with how we assess the quality control. Not only we have this client tool we call AutoQC, and one of the things we felt like we had a pretty good strength is we have this ability to interface with the instrument, import data into a client tool, and then upload it to a server. So one of the things we thought would be an excellent capability would be to have the instrument periodically collect certain measurements and be able to track that longitudinally, so we can use it to track the performance of either an assay or actually how the system was performing over time. So just to kind of show a little bit of this, is in general, we tend to think about system suitability, and in our lab, we think a lot of this is kind of process controls. There's also, of course, quantitative quality controls as well. But what we do mostly for proteomics, we track whether or not the system is performing well and whether or not a given sample preparation worked well. And so in one case, we use a set of peptides and a protein digest that we just inject every certain number of injections. And in another case, we actually add certain things into the sample that we use to track whether or not the performance of that specific sample is working okay. So one's just used to assess the system, and one's used to assess the sample prep. So the system suitability is kind of basically a sample that doesn't change. It's similar to your actual samples. It doesn't really matter what it is. It's actually just something, if you're making an assay, you want to be able to track that over time longitudinally. It should assess, whether or not the different parts of the instrument's working okay, like mass accuracy or chromatography. And we, in general, we use a very simple, commercially available set of samples. And we definitely assess different parts of these very complicated instruments. And we use things like control charts, and we can use things like Westgard rules to be able to flag different portions of the performance. We have this little client tool that sits on the instrument. It kind of logs people in and uploads new data as it's coming off the instrument in real time. We can track all of these things longitudinally. We can see whether or not there's going to be outliers. We can look at different ranges of peak areas, mass accuracy, et cetera, over time. And you can begin to annotate things like if you're doing instrument maintenance. These instruments cost millions of dollars, and what we were kind of shocked at was that there were no real tools that the instrument manufacturer were providing that allowed the instrument to be tracked longitudinally over time. And each of the labs that did do this fairly well had become like they had set these very ad hoc ways that each lab had used to assess the performance of the instrument. And we wanted to come up with something a little bit more generalizable that the community could use. This has been one of the largest adoptions of our platform, has been just this ability to track things in the parts of the system over time. We have this dashboard. So this is just kind of showing 8 instruments in our lab. Our lab not only makes software, we're also an analytical lab. And there are, at the time I took this snapshot, there were 3 instruments that were actively running and collecting data at that time. And you can kind of see all of whether or not there were outliers, whether or not there were different performance metrics that were being tracked, et cetera. We can see that this was actually indicative of whether or not our product ions were no longer giving good signal. We had, a cylinder on one of the instruments had kind of run out. We weren't getting good fragmentation, so that showed that that instantly triggered a response. We also can see that in general, the mass accuracy instrument's been pretty good, but there was this time here within about a 3-day period where the mass accuracy got a little bit worse by about 2 parts per million than normal. We can also track other things like retention time, and this is where we happen to have a column clog, and it caused instant triggering that there was some performance issue. We tend to have these portions within the PanoramaWeb server. Each lab or project has their own system suitability. It's a lab-wide folder that we tend to think about, and this tends to be something that we encourage people to set up in this way and allows people to track, just performance of the system over time. The process controls tend to be a little bit more of an experiment-wide type metric, but we use the same pieces of software and visualization, but we just track different samples and different matrix. It allows us to assess whether or not the sample preparation had gone bad or there's some other issue that occurred. So basically if something happened to the sample prep or something happened to the cleanup and recovery, you tend to get different responses between different portions of the things that were added into the sample. Again, the software is very flexible. It just allows you to track different analytes across time and allows you to be able to provide annotations for that. And this is just a way in which our lab has done it, but it can be set up many different ways. So one of the things that our lab also does is we tend to set up a folder for every experiment that's happening in the lab. And the software is, again, extremely flexible. It allows you to have different wikis. So you can add things like metadata for the different projects, and these can go into relational database tables. You can also have wikis to describe the experiment or methods or protocols and keep all of that in one continuous place. There's a lot of versioning that's handled with it, so different people can edit that document. And you can have subfolders. In our case, we may have in most cases, we have 2 types of subfolders within each experiment. One that we track the process control, which gives us a sense of how good the data is. And then we have another folder, which we track actually the quantitative data. So in this case, we showed that some of our process controls started to fall outside of our normal acceptable criteria in this experiment. It was a pilot experiment that were being done. The area in the gray portion is where we kind of set the instrument as being control and set these boundaries range as being this is what should be normal. And there was clearly outside that range for some portions of the measurements. And then we also have this folder where we actually store our quantitative data. So in there we have the documents, the raw data files, all stored basically in one location. It's easy to go back and find it. We've been able to search and add queries to this, and so when people or trainees or students or staff leave, the data is somewhat stored in a reproducible location. I'll point out this server is not just for our lab, it's used for 547 labs currently. And you can, here is a Skyline document. This is actually, again, a project that's been associated that we're with some cerebral spinal fluid analyses that were being done. We have a series, and you can drill into the data depending on how deep you want to go. So one of the projects that we are working on with the NIDDK is this effort we call TaMADOR. TaMADOR is it's meant to stand for Targeted Mass Spectrometry Assays for Diabetes and Obesity Research. In order to, this is a collaboration between 3 NIDDK funded grants. One at the University of Washington in the School of Medicine here. Andy Hoofnagel's lab is developing assays specifically for type 1 diabetes. And we're collaborating closely and interactively because there is definitely some overlap in the types of assays that are wanted by our clinical colleagues in making assays for specifically obesity. And we're working with Jenny Van Eyck's lab at Sinai in the design and validation of assays for obesity, but also Wei Jun Yan's lab at Pacific Northwest National Laboratories. So in order to support this project, we set up a web page on Panorama. We can get custom URLs out of it. We can also control things like message boards. This is, again, there was no code that needed to be written for this. This is already part of our software that we provide. Any group that wants to work on this can actually do this. The control of the security for internal resources, agendas, meetings, logs, etc., are all stored within this this web page. You can click on different portions of the links, and for instance, here is you get some description about the abstract of our grant here at the University of Washington, and you also get some subfolders that provide and store and share managed data for our project here. You can kind of drill a little bit deeper. One of the things that we kind of did as a joint project was took an assay that was developed at the University of Washington to measure insulin C-peptide. We had a full detailed standard operating procedure. And then from there, we were able to make a website that had wiki, some sort of description of the assay, a little bit of what the data should look like within the Skyline client tool, what the calibration curve used from one of the multiple days in validating the assay. And then this could also be shared with the groups in the larger consortium, and then we can get data back from those groups as necessary in different subfolders and make it more broadly available. So someone can go and drill in and look more closely at the underlying data. I know this is not a mass spectrometry group, but this is an exciting thing to be able to do is to be able to not only have a client tool that works with all 6 major instrument vendors, but also you have a server-side application that people can go and see the data without necessarily having to interface with the client app. The client app definitely does have more feature-rich capabilities, but at the same time, often people just want to be able to see what the underlying data is and what an example of that assay should look like. It doesn't just support this TaMADOR project. The TaMADOR project is the main project we're working on with the NIDDK on. We also have a project that's supported by the National Institute of Aging, and this is looking at assays more for Alzheimer's disease and dementia. And again, this is a multi-project consortium, and we kind of set this up in the same sort of ways which we established the web page for the TaMADOR. We've been involved in some common fund projects. Here's a very large project in which there were like thousands of assays that were performed over the period of time. It was done in collaboration with Jake Jaffe. And again, this is kind of the endpoint and place where people share the data and make it available. And there's a lot of flexibility for how people can establish their own website on the server. We also are the back end to a lot of NIH branded sites. So for instance, the National Cancer Institute has what they call this assay portal. And they have their own NIH branded site, but they pull the data and the information from our server and they display it on their server. This has been something that a lot of groups have begun to make more use of, and we found that different labs like to take advantage of that. And this is, I think, particularly powerful way that labs can maintain their own branding or their own and their own identity, and they can design a website in a way outside of it, but at the same time, they don't have to think about storing mass spectrometry data, which is this incredibly complicated way of managing results. As long as the data is in a format that's compatible with our server, such as the raw data files and also the Skyline documents, which is the process results, it makes it fairly easy for us to be able to serve this up with all the full security through application programming interfaces. We also have this place whereupon we've worked closely with the journals and also with the funding agencies to come up with ways to share data upon publication. These are each of these different rows of this table is basically data associated with a different publication. And when the publication actually becomes fully known and available, that link is provided. We have what we call proteome exchange IDs, which are the unique identifiers in the mass spectrometry and proteomics space that we use. We can also assign digital object identifiers. We can also provide licensing terms for different projects. And these are things that we found to be extremely useful. It's all searchable by author or organism or instrument, some certain metadata. And we're constantly trying to improve this. We do feel like the use of metadata may not be the ideal currently. People can, when they go to share data, they can provide information about the paper, their data, the process results, how they can add figures to it. They can get their own custom URL that they can share in their paper. It can be kept private, where they can get login information that's provided to the journal for reviewers, or they can make it fully public at that time. And the nice thing about it is that it's fully immutable to the data producer. This is something that's a requirement for a lot of the journals. They don't want people to go back after the fact upon published data and change the results. So we maintain an extra copy of it. That copy becomes completely immutable. One of the things that we're pretty excited about, one of the things that the mass spectrometry and proteomics and metabolomics fields have been struggling with for a long time is ways of tracking samples and freezer logs from repositories. And over the last 2 years, we've spent a lot of time trying to integrate another platform that our colleagues at LabKey Software have developed, which is this sample manager and sample tracker. And we've basically built another interface or module on top of that, which is our Panorama module. But what we'd like to be able to do is, if you had samples that were then analyzed with a Skyline document, there would be links back to that individual sample, back to which Skyline documents that is present within the Panorama server. So the plan in the future is also this ability so that we can use the metadata stored in this sampled manager and be able to link this information back more broadly. So we do have this a little bit more of a broader vision. This is something that's definitely an ongoing project. And this vision has been to basically host all information about sampled metadata, the preparation location, use that information to design experiments. We want to be able to track the system suitability and process controls over time. We want to be able to add metadata to the Skyline document from the relational database. We want to be able to, process, of course, right now, most of the analysis are done on each person's client computer in the client application. But some of these experiments tend to get a little bit computationally expensive. So we want to be able to handle that more in kind of a cloud or a workflow type manager for computationally expensive steps. We want to be able to manage the process results, so things like audit logs, version controlling, et cetera. And one of the things I think that's fairly unique about this, at least in the mass spectrometry space, is how much flexibility there is in the user-controlled access. And we also have worked really hard to make the mass spectrometry and the quantitative data findable upon publication. And this is largely done within the community using Proteomics exchange IDs, but also by searches through Panorama public website, and also digital object identifiers, et cetera. And also, if you have samples that are within the sample manager, you should ultimately be able to find the analyses and the results that were performed on the individual samples. Just to provide a little bit of an acknowledgement, I know that this is a little bit outside the area that normally is hosted by this webinar, but the people that in my lab who have been working predominantly on the Skyline and Panorama project are shown in red, and Nat Brace, Rita, Brian, Brendan, Brian, Vigisha, Nick, and Kaipo are either project managers as far as dealing with the training or dissemination or tutorials, et cetera, of these software tools. Brendan McLean is the project lead of this. He's the software development manager of the effort. Vigisha Sharma is the Panorama lead. So she's one specifically that's been working in kind of designing and implementing some of the aspects of the server in close collaboration with the other people that are shown in red. This has been a detailed collaboration with LabKey software. Most of the software that's developed by LabKey is open source. They now have some enterprise pieces of software that do require some licensing, and they also work very closely with a lot of our pharma colleagues. So where they provide kind of some custom engineering to improve the development of Panorama funded by the pharmaceutical industry. A lot of this effort that's being done and presented here today was done very closely with Andy Hufnagel's lab and part of this TaMADOR consortium, which is an NIDDK funded effort. A lot of our other clinical type analyses are performed very closely with our pathology colleagues at Stanford University. We have, of course, a lot of fantastic former lab members who have worked on these projects and others. And really thankful, and we're very fortunate to be able to have NIH funding efforts to support the development of these tools. And again, I've mentioned that this is a partnership with the instrument vendors as well. We are kind of one of the primary tools that interface with all 6 instrument results, and also helping build instrument methods and kind of have this sort of the cyclic process by which we can build new methods, analyze the data, share and organize those results. And I'm happy to answer any questions about this project or any of the other things our lab does.
Okay, thanks, Dr. MacCoss, for the presentation. If you have any question, now is the Q&A session. You can either unmute yourself or type the question in the chat. And then I think during the presentation, probably from the previous slide, there's one question from the audience. Could you show me again how to find mass spectrometry data generated from a paper, in a paper from the homepage? Yeah, because I think, yeah.
Yeah, so in most cases, what will be provided is this during publication is what's called a proteome exchange ID. And so with that proteome exchange ID, let me just share my my screen again. There's these proteome exchange IDs, and these are all searchable. And also you can go to the proteome exchange website. So our server is one of 7 that the community has kind of established, and all of the 7 proteome exchange servers all kind of serve slightly different functions. So you can actually go to the proteome exchange website and you can type in and that proteome exchange ID. It will then take you to a link, which will then take you to the data site on the respective individual servers. I hope that answers the question, but that's kind of the common way. All of the 7 proteome exchange servers have slightly different mechanisms of storing and managing the data, and they all have they slightly different types of data that they display. Our server is very focused on quantitative data that's been processed with a document called our Skyline client. And that's kind of one of the prerequisites that we kind of require people to have when they share data by the Panorama website.
Thank you. There is another question. Probably it's also this page. So could I find a proteomics experiment or assays through typing in relevant keywords such as project name or disease name, other than the protein of interest?
Yeah, currently, no, that's actually a good point. So with that metadata is not stored. We've been having discussions with the Proteome Exchange to actually expand the metadata fields. We have a separate, I've been a consultant on this, on an effort called the Proteomics Data Commons, which is being done, handled through the National Cancer Institute. And there, the clinical metadata is much more extensive. One of the problems that we have, as everybody knows, is getting people to enter in extensive clinical metadata in a common vocabulary. And what most people end up doing at the time of publication is just trying to fulfill their journal publishing requirements. And a lot of people don't have significant resources in order to annotate their data files with the same appropriate common vocabularies. So we've kind of erred on making it simple and easy for people, but at the same time, the level of metadata that we actually have available has been probably is not ideal in all cases. And I'll point that that's not just an issue with our server. That's actually a problem across all of proteomics data sharing and data exchanging sites. Right now, there is people end up, the easiest way that people find a data set is because of by reading the publication and then going back from there. There's been less effort to make it so people can query directly for a specific disease type or application. That's definitely something we'd like to be able to improve more in the future. There has been a lot of discussions about formats and types of ways that people can annotate this and what sort of common vocabularies to use. I think.
Okay, thank you. There is one more question. I think, what is the best way for me to cite the experimental essays from people who shared in Panorama to the publication or when submit a grant?
Yeah, so the way that the field has kind of adopted is the Proteome Exchange ID. It's just to acknowledge it, and people can either just that's the best way to acknowledge it. The most of the licenses that people assign, so each of these publications are assigned a license, and what most people choose either is CC0, so Creative Commons 0, which is it's in the public domain and they don't require any attribution, and then or their CC BY, which is they require attribution or they would like to have attribution. It's not enforced, obviously. It's really hard to enforce this, but in those cases, people do like to have the Proteome Exchange ID at least recognized either in the publication that data came from that source. So the Proteome Exchange is the most common way that people share their information. Some journals do like digital object identifiers, and people can request those at the time of submission, and we will assign a DOI. It looks like there was a hand up.
Yeah, so I had a great presentation. I had a quick question, and you mentioned sort of the tracking of samples and things of that nature. Are you doing any work in terms of, when data gets, data sets sometimes contain, multiple types of data. So if data, for example, has to go to geo and then other places, there is the NCBI biosample. Are you working with them as well in terms of sample identification and integration with those types of resources?
Nothing that's really formalized. It's more been as people have come to us with a specific project and they say, we have these samples, we need to kind of link it to this existing data set. We've kind of done it as people have come to us project by project. We spent a lot of effort within the Links project, which was a Common Fund project, and there was a data integration center that was very focused on making sure all the metadata was standardized across all the sites. So in that case, their website was provided lots of links back to their processed mass spectrometry data on our server. They also made it so that they can pull actually the underlying quantitative data from our server. And we also linked our server back to theirs, so where you can get all the full metadata as well. So it has been these very one-offs. We are working with on a separate effort with the National Cancer Institute, where we've been focusing on clinical specimens, and some of those, in those cases, they were analyzed by the TCGA, and they were provided a little bit differently as well. So those are samples that are stored in the genomics data commons as well as in the proteomics data commons, and there is an effort to try to link them. It's been fairly early on though. I think the problem is there's no standard, right? And then it seems like every major project has a slightly different standard or different.
Yeah, and there is a question from Andy. Yes, it's a question for the dkNET team. Should Panorama be a suggest data repository for proteomics?
Yeah, so Andy Hoofnagel is actually the PI of the project here, and I think his point's quite valid. As far as the dkNET goes, and is there any way that we can work together in that regard?
Sure. Yeah, I will take a look. Yeah, I think it will be good for us to including. Yeah. Mhm.
We definitely have, as I said, websites within Panorama for specific DK projects, right? So there is this TaMADOR project, and maybe we can find some way to link them.